why use Caenorhabditis briggsae instead of its more popular relative C. elegans? One of the reasons lies in the phylogenetic tree shown in the upper left-hand corner. You can also see the world map in the bottom right-hand corner with asterisks representing the locations of some of the many natural populations of C. briggsae that have been collected from around the world. For example, VT847 on the very far left from the Hawaiian Islands. So C. briggsae is a globally distributed species like C. elegans, but its phylogeny is quite different from C. elegans. And that is that if you collect and compare the genotypes of populations from the tropical circles of latitude, shaded in red, they form a distinct clade at the top of this phylogenetic tree, where any population from a tropical circle of latitude location on the planet is more closely related to each other than any population collected from the temperate regions, shaded in blue, and from the equatorial regions, shaded in yellow. The tropical circles of latitude clade, in red, is on average about 1% genetically different from any temperate population, shaded in blue. And this is particularly curious because a common pair of populations that are used in genetic analyses, including looking for genetic incompatibilities, is the population pair AF16 from India and HK104, which was originally isolated in Japan. They're about 1% genetically divergent, which is about the same genetic distance as humans and chimpanzees, our closest relative. But look at the difference, for example, from HK104 to a different temperate population, PB826 from Ohio, United States. They are very closely related to each other in the phylogenetic tree in the bottom, but they're half a world apart geographically. So this phylogeographic structure in C. briggsae raises a lot of interesting questions that geneticists and evolutionary biologists study. The key is that there's so much genetic divergence between the blue and the red shaded populations, the temperate and the tropical circles of latitude populations, that there are lots of genetic incompatibilities that are starting to develop between those blue and those red clades. And that gives us the opportunity to look for differences that might cause things like hybrid incompatibilities. Therefore, looking at the genetics of speciation. C. elegans, again, does not have this population structure, and most of their populations are much more closely related to each other genetically than the red and the blue populations here. So C. briggsae seems to be a better model organism for studying genetic divergence and its effect on reproductive isolation and speciation.